I think probably the most exciting thing we have this year is 2 sessions on animal optics. One that's talking about the design of animal eyes, and so we have some exciting sessions on the design of those eyes, polarization properties in those eyes, how multifocal optics in various eyes, and looking at fish and other species that are really exciting in the evolution of how they've developed. With the idea that knowing the design of the animal eyes might actually help us in developing optical design in other applications and other lens types. So that's the one exciting session, and the second session that runs right after that is a session on imaging and disease models of animal eyes and how we're using animals and imaging techniques to start studying diseases such as the development of nearsightedness, known as myopia. We have Melissa Gang speaking on the imaging of a mouse model with adaptive optics, and she's got some great images looking at ganglion cells. We're looking at development in guinea pig eyes, which looks also really exciting. The idea is to understand more about myopia development in the guinea pig model and then translate that into looking at it in humans and understanding maybe how we could correct it in humans, why we become nearsighted. And so that's exciting. And of course the mouse models, they're great because we can use transgenic models in mice to start studying various diseases and looking at treatments then as well, which is exciting. I'm actually most excited about one of the symposia. We've been working on developing the symposium for the 50th anniversary of the Smirnov's aberrations measurements, and so that's really exciting because without that we would never have gotten to the point we are now, where we're using aberration correction to study vision, where we're using it to develop multifocal, or looking at intraocular lenses and how those and aberrations can play a role in that, how aberrations play a role in refractive surgery for eyes, so LASIK, custom LASIK comes out of that measurement. And then we also use the correction of those aberrations to study visual processes, to correct the aberrations for retinal imaging. And so that's actually allowed us to see single cells at the back of the living eye and study how they're connected in terms of neural processes, and looking at that, how they're connected in terms of functional measurements, and just looking at structure. So that's been really exciting. So I'm excited to see that symposium of sort of the history of the aberration measurements, and then looking at what we've been able to do now that we've been able to measure them, and now we can use adaptive optics to correct them. One of the other sessions we've got going this year is fluorescence imaging and other techniques. We have invited speakers in that area talking about how to get fluorescent markers into the eye, into living cells. Ken Greenberg is going to talk about new techniques that he's using to get viruses and vectors and into the living eye so that the cells will actually express fluorescent markers, and we can go in then and image them either in vivo or ex vivo, looking at the processes and how they're affected, and even potentially some of those looking at functional and giving us some changes there.